Hi everyone, today we're going to be talking about DNA. We'll start off with a bit of an introduction and then we're going to look at the structure of DNA. If we talk about a chromosome is going to be composed of DNA along with packaging proteins. The DNA is the portion that contains the genetic information. If we look on a strand of DNA, we'll find different genes, and these genes are what is controlling inheritable traits that we see. Generally, genes control the production of proteins, and it's those proteins that are bringing about the trait that we're seeing. So let's have a look at the structure of DNA. DNA is a type of organic molecule known as a nucleic acid. The monomer of nucleic acids is a nucleotide. If we look at the structure of a nucleotide, it is composed of a pentose sugar, pentose meaning that it contains 5 carbons. It also has a nitrogenous base attached to it, and then a phosphate group. When we're forming DNA, the first thing that happens is we attach a number of nucleotides together to form a polynucleotide chain. These nucleotides are attached to one another using covalent bonds. The bond joins the phosphate group of one nucleotide to the sugar of the next nucleotide in the chain. DNA is double-stranded, so that means we have 2 polynucleotide chains attached to one another. The second strand of nucleotides runs in an anti-parallel direction to the first strand, so that the bases are pointing inwards. Once again, the nucleotides in this polynucleotide chain are attached to one another using covalent bonds. The 2 polynucleotide chains are connected to one another through hydrogen bonds. We indicate hydrogen bonds with a dotted line to show that they are weak bonds. It is important that this is a weak bond because we need to be able to separate these 2 polynucleotide chains so that we can read the sequence of bases on each strand. The nitrogenous bases in DNA are adenine, guanine, cytosine, and thymine. There are base pairing rules, and these rules state that adenine will always pair up with thymine, while cytosine will always pair up with guanine. Let's say, for example, the sequence of bases on this polynucleotide chain was adenine, cytosine, thymine, guanine. Using these base pairing rules, we could then figure out what the sequence of bases is on the strand that is bonded to it. For example, if this is adenine, that would make this base thymine. If this is cytosine, then this base would have to be guanine. If this is thymine, this one would be adenine. And if this is guanine, then this one would be cytosine. When looking at the structure of DNA, the 2 polynucleotide chains would wind themselves around an imaginary axis, forming a helical structure. We call this the DNA double helix. Each chromosome will consist of 1 DNA double helix along with packaging proteins. Each DNA double helix will contain many genes, and each gene will control at least one distinct trait. So just to recap, each nucleotide is going to be composed of a pentose sugar, and in the case of DNA, that sugar is deoxyribose, hence the name deoxyribose nucleic acid. The nucleotide will also contain a phosphate group as well as a nitrogenous base, and in the case of DNA, our nitrogenous bases are adenine, thymine, cytosine, and guanine. We have base pairing rules where adenine will always bind to thymine and cytosine will always bind to guanine. DNA strands are complementary. That means if we know the sequence of bases in one strand, we can easily figure out the sequence of bases in the complementary strand using our base pairing rules. So for example, if we had the sequence of bases in our one strand being GA, GACT, what would the sequence of bases in our complementary strand be? If we have G, we would have C, If we have A, we would have T. G would be C, A would be T, C would be G, and T would be A. Genes are going to differ from one another in terms of that sequence of bases. They may also differ from one another in terms of their length. The term genome refers to an organism's complete set of DNA, including all of the genes. This means that the genome contains all the information needed to build and maintain an organism. Each species has a unique genome. If we look at the human and the bonobo chimp genome, they are 98.7% identical. That means that a difference of 1.3% on the genetic level brings about this enormous difference we see in the phenotypic level. Alright, now's the time to go ahead, pause the video, answer these review questions, and when you're done, we'll come back and we'll go through the answers together. Okay, so the first question, what two types of bonds are found in the DNA double helix and describe what they're joining? Well, the two bonds we see are the covalent bond and the hydrogen bond. The covalent bond is joining nucleotides in the same polynucleotide chain, connecting them via the phosphate group of one to the sugar of the next one in the chain. Our hydrogen bond, on the other hand, is connecting our two polynucleotide chains to one another, and this is done at their bases. What is a genome? Well, the genome is the complete set of DNA, including all of the genes that is found inside an organism. What is the percentage of thymine in a DNA double helix in which 20% of the nitrogenous bases are guanine? Well, to answer this question, you need to remember your base pairing rules. Remember that guanine is going to bond with cytosine. So if we have 20% being guanine, that means we also have 20% being cytosine, which means that 40% of that DNA double helix is taken up by our GC bond. This leaves 60% to the AT connection, which means that if we're looking at how much is thiamine, it would be half of that, which is 30%. For the final question, we needed to find the complementary sequence of DNA to the following sequence. All you need to do once again is remember your base pairing rules. So if there's an A in this strand, the complementary strand would have to have a T, thiamine. If thiamine is in this complementary strand would have to have adenine. Guanine, complementary strand has cytosine, T-A. Cytosine in this strand means our complementary strand would have guanine. Once again, another guanine, and then A would be T for thymine. In our next video, we'll look at how this information in DNA is used within the cell to create proteins.